So unlike the macromolecules that we've been talking before, where there's a common monomer that makes up the polymers, such as nucleotides make up nucleic acids, amino acids make up polypeptides. For lipids, there isn't a monomer that is common to all types of lipids. One thing that is common to all lipids is they are hydrophobic, and that is due to the fact that lipids will have a very large hydrocarbon component. The different types of lipids, fats, steroids, phospholipids, waxes, the most important to, from a biological standpoint, that is what the textbook talks about, are the fats, steroids, and phospholipids. I will mention a little bit about waxes. And the function of lipids is very diverse depending on the type of lipid, but they can be used anything from the source of energy that we most think of when we think about fat, and but they are very important component of the cell membrane structure, and they can actually form structures. You wouldn't think that a lipid a structure can be composed primarily of lipids, but it can. You will find out. We'll start with fats. Fats are called triacylglycerides. They can also be called triacylglycerols. Same thing. And here you have common to all lipids, hydrocarbon chains, which is highly hydrophobic. And this is, when you think of fats, this is the structure of it, and it is involved in a source of energy. So for the triacylglycerols that I showed earlier are composed of fatty acid chains. So this is a fatty acid chain that is attached to a 3-carbon backbone. Okay? And this backbone is glycerol. Okay? So it's a 3-carbon carbohydrate. Okay? And the hydroxyl group, one of the hydroxyl groups, will react with the carboxyl group of the fatty acid. And this is a type of dehydration synthesis. And each of these hydroxyl groups serves as an attachment for the fatty acid. So you have 3 fatty acids, tri, and the glycerol backbone, hence the name. The fatty acid chains that are attached to the glycerol backbone can be identical or similar, but so they can be all the same, or they can have differences in the fatty acid group. So you've heard of saturated and unsaturated fats. What's the difference? It has to do with the presence or absence of double bonds. So the saturated fats, here you have an example of a saturated fat. Here's our 3-carbon backbone, the glycerol backbone, that has the fatty acid chains. And the fatty acid chains are just represented in a zigzag line. Each of these joints represents a carbon, and they don't even bother writing the C for the carbon or the hydrogens. It's just assumed to be there. And these carbon-carbon bonds are single bonds. So there's no double bond, as you can see here, between the carbons for any of these 3 fatty acid chains. And you can see from the more 3-dimensional structure that it forms a linear chain. So the lack of these of double bonds, so only single bonds, explains why saturated fats will be solid at room temperature. And it has to do with the fact that if you imagine that here's my 3 carbon backbone, and these represent each of the 3 fatty acid chains, so glycerol backbone I've held together, and the 3 fatty acid chains. And because of the structure of the fatty acid chains and the fact that they are planar, I think I used the term linear before, but they're planar, they can stack on top of each other, those 3 fatty acid chains, as so. And that explains why they tend to be more solid at room temperature. And here we have a solid set of saturated fatty acids, saturated fats, composed of saturated fatty acids. And I might have shown you this already, and you'll see this in the PowerPoints a lot, this URL. And it's a very nice way of showing the structures. And they have each of the 4 macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And if we go to, here's our fatty acids. So here, if you click on these, it'll bring up the different molecules. And here's palmitic acid, which is a type of fatty acid. It has a carboxyl group, C double bond O, OH. Here are the carbons in gray and the hydrogens in white. And you can see that the 3-dimensional structure, if you look at it from a space-filling model, see how they're planar. And let's compare it to an unsaturated fatty acid, like oleic acid. This has an 18-carbon backbone, and here's our carboxyl group, not carbonyl. It's not C double bond OH, it's C double bond OH. And you can see the hydrogens shown here. And so carbon has 4 bonds. It has, here this carbon has a bond between this carbon, so that's 1, 2. Here's a hydrogen, here's a hydrogen. Okay? And note that this carbon has 1, 2, 3, 4, so the maximum. There's no double bonds. But notice here this carbon has 1, 2, 3, 4, but this carbon has 1, 2, 3, 1, 2, 3. Where's the 4th bond? It's a double bond right here. And same with this carbon. 1, 2, 3. Where's the 4th bond? It's a double bond. It's the double bond. Okay? And you can also notice that there are fewer hydrogens attached to these carbons because instead of hydrogens, you have a double bond. So here, each carbon has 2 hydrogens, 2 hydrogens, 2 hydrogens. But here, it only has 1 hydrogen, and that's because the other bond is a double bond between each of these 2 carbons. And if you look over here, you can even color the double bonds, and you can show the double bonds also like this. This 18:1 refers to the fact that it's an 18 carbon with 1 double bond, and the delta 9 represents the placement of this double bond on the molecule. So placement, or better said, location of the double bond. And so the 9 represents the fact that here's the carboxyl group with the 1st carbon, 2nd, 3rd, 4th, 5th, 6th, 7th, 8th, 9th carbon and the 10th carbon is involved in the double bond. If you clicked on close view, you can see in greater detail the double bonds. If you click on linoleic acid, which is another type of fatty acid, here's the carboxyl group, and it has it's an 18 carbon with 2 double bonds at the carbons 9 and 10, and 12 and 13. So reading from the carboxyl group, here's carbon 1, 2, 3, 4, 5, 6, 7, 8, 9. Notice that there's only 1 hydrogen instead of 2 like the others. So this is involved in the double bond, so carbon 9, 10. 11, 12. So carbon 9 and 12 are involved in the double bonds. And similarly, if you look at linoleic acid, it has 3 double bonds. So look at the 3-dimensional structure shown as these stick models of the different types of fatty acids. So you have your saturated, which is very planar, and notice that when you introduce a double bond, it will change the planar structure to a more kinked, structure. And the more double bonds, the more kinks in these fatty acid chains. Doink, 3. Okay. And how does that change the overall shape of the fat if it's saturated versus unsaturated? How does that explain why unsaturated fats tend to be liquid at room temperature versus solid, like the saturated fats? So here's my handy dandy triglyceride or triacylglycerol, okay, 3 carbon backbone, and the 3 fatty acid chains, 1, 2, 3. And so these 3 fatty acid chains without a double bond, okay, so they're saturated fatty acids. If you introduce a double bond, or maybe 2 fatty acids have a double bond, or maybe one of the fatty acids has 2 double bonds. Notice how it changes the overall 3-dimensional structure. And this does not stack as well when you have the saturated fats, and as a result, they will be more liquid at room temperature because they occupy more space. Yeah? Just like how if you crumple up paper, I'm going to do it. You can't fit as many of these in a box compared to a box of the flat paper. I think you get it. So if you go back to the home page, you can see, we just saw the fatty acids, you can see the triacylglycerols. So here you have 3 carbon glycerol backbone with the 3 fatty acid chains. And the full name of this fat takes into account in the name each of the 3 different fatty acid chains. Okay? So you have your glycerol right here. You have palmitic acid, the fatty acid, which is in the name. And it's the 16 carbon with how many double bonds? 0. Obviously, this one has the oleic acid has 18 carbons. The stearic acid has 18 carbons, but the oleic acid has a double bond. And you can tell if you turn the structure, you can see how planar this this fatty acid chain and this fatty acid chain are. And the center fatty acid chain, you can see that there's the double bond between the carbon, this carbon and this carbon. And if you count down, you can see that it's on carbon number 9. And if you want, you can change it to a space filling model and you can see how it would occupy more space than a fat that has a triacylglycerol that has 3 fatty acid chains that are all saturated. This one of the fatty acid chains is unsaturated. So back to our PowerPoint. Here it's showing that this fatty acid chain has the double bond, and it introduces a kink. Notice that it says a cis double bond. Okay? Cis as opposed to trans. And by the way, here's the URL for that website we were just at. Okay? And again, because they cannot pack as densely, it will be liquid at room temperature. Now, what does it mean that this double bond is in cis? And does that have anything to do with trans? And when we're talking about trans fats? Yes, it does. So, through the hydrogenation process, we can take this lovely liquid, fat, okay, from plants, and we can turn it into a solid fat. Now, why would we want to have a solid? Take this nice vegetable oil that has a lot of unsaturated fatty acids and make it hardened. Okay? Well, in the morning, you want to, say, spread butter on top of your toast, and butter is solid at room temperature. However, butter is derived from animal fat, and animal fat also has cholesterol, whereas plant fats do not have cholesterol. Plants do not have the enzymes necessary to make cholesterol. So plants are a great source of fats that in the end you won't get cholesterol added. However, some people need to have a low cholesterol diet, and so instead of being able to have butter on their toast, people that needed to have lower their cholesterol in their diet couldn't have this nice solid fat on their toast. So what to do? How do we make a fat that is solid at room temperature but doesn't have the cholesterol? Well, instead of having it from animal fat, where you have saturated fat, why don't we take this plant fat and instead of having the double bonds, which will make it liquid, remove the double bonds? How? By hydrogenation. And so they actually add hydrogen gas to the liquids and introduce the hydrogen so that there's no more double bond. And as a result, the fatty acids that were unsaturated become saturated and solid at room temperature. Now, so again, turns the unsaturated oil into saturated, and so when you do that, this saturated fat is in the form of margarine. Lard is the same idea, plant produced. But trans fats. Trans fats are an unintended byproduct of hydrogenation. So here's part of a fat where it's just showing one of the fatty acid chains and taking a close-up of, this is a fatty acid chain of an unsaturated fatty acid. Okay, so you have the double bonds. And notice that the carbons that are in the double bond, the hydrogens are in cis. So cis basically means same. Okay? So here you have your carbon-carbon double bond, and when carbons are, maybe I'll put it this way, when carbons are involved in a double bond, it makes that region of the carbon-carbon double bond highly planar. And the hydrogens because each carbon in the double bond will have only one hydrogen. The hydrogens can be on this carbon. I'm going to have to show you in a picture. I'm going to have to do this an old-fashioned way. So here you have a carbon-carbon double bond. And I haven't shown the hydrogens yet. Presto changeo. Okay, I've added the hydrogens. And those are the hydrogens that are in cis. All right, so cis double bond. But there's another orientation. Ta-da! Presto changeo. The double bond, the hydrogens can also be in trans. Okay. So you can have the carbon-carbon double bonds with these hydrogens in cis or trans. Now, remember the hydrogenation process will take this double bond that makes oils liquid at room temperature and hydrogenate and reestablish the saturated. You can think of it as saturated with hydrogens, because there's more hydrogens on this molecule than there were here. Okay, so here there's 2 more hydrogens than there were in the unsaturated. Okay, so here we have a saturated fatty acid, and that's via hydrogenation. But an unfortunate byproduct of this hydrogenation process is that instead of forming a saturated fatty acid, it remains unsaturated. However, the hydrogens are in trans. Okay, so cis, trans. Okay, cis, same side, trans. Okay. Now, even though this is unsaturated, it's a trans unsaturated fatty acid. Here's the problem. This can cause coronary heart disease. And are trans fats as dangerous as big of a health risk as made out to be in the literature and in advertisements? Are trans fats as bad as they seem? Oh yeah, they are a serious health concern. Okay? It causes things like coronary heart disease. So here is a more three-dimensional structure of the unsaturated fatty acids that have the hydrogens in cis versus the hydrogens, here's the double bond, in trans. Unintended byproduct of hydrogenation, and unfortunately has some very serious health effects. So having trans fats in your diet, in addition to saturated fats and having cholesterol, will raise a type of lipoprotein called LDL. Okay? So if you've ever heard of good cholesterol and bad cholesterol, the LDL, low density lipoprotein, so it's a lipid with protein molecules attached to them. This, the saturated fats, trans fats, dietary cholesterol will increase low-density lipoproteins in your blood and risk coronary heart disease. So here's your heart. The coronary, they're talking about the coronary artery that feeds blood to the heart. Kind of important. And the trans fats will decrease the HDL, which is the high-density lipoprotein. The high-density lipoprotein is what you want. Trans fats will reduce it. So on a blood test, if you ever get your cholesterol levels done, it will tell you the HDL versus LDL ratio, and you want to have a high ratio of HDL and a lower ratio compared to the LDL. Okay. HDL, good. LDL, and now it's time for steroids. In lipids, there isn't one monomer that is common to all types of lipids. So steroids are one type of lipid, and they have one thing in common to all steroids. They have a 4 rings and then various functional groups attached. So here is cholesterol, a 4-ring structure, 3 6-carbon rings and 1 5-carbon ring. And here's a natural anabolic steroid, where it has, again, the 3 6-carbon rings and a 5-carbon ring. And they can have various, again, functional groups attached to them. Steroids have a lot of important functions. Cholesterol is a type of steroid, and cholesterol is often found in membranes. Bile salts, cholesterol is in biosalts. Bile salts are important in digestion. Steroid hormones, I'm sure you've heard of the sex hormones, estrogen and testosterone. And all of these have this 6-membered ring and a 5-membered ring fused together, and various functional groups attached. A lot of our hormones are derived from cholesterol, which again is a type of steroid, and steroid hormones include things like cortisol, which is an important anti-inflammatory, and vertebrate sex hormones, like I said, testosterone and estrogen. Cortisol, of course, is also one of the what they call stress hormones, and will be produced under stressful conditions. But cortisol is also an anti-inflammatory that is an active ingredient in Cordate, which is a topical anti-inflammatory. So steroids are a natural molecule that the body produces, and when we talk about the sex hormones, such as testosterone, the body will produce testosterone naturally, both in males and females, but at different levels. But sometimes people can abuse anabolic steroids, specifically testosterone, and it can have some serious consequences. So bodybuilders will sometimes abuse anabolic steroids, and people in sports, and the consequences are severe and often irreversible. I had my dad, I grew up in a vet clinic. My dad was a veterinarian, and occasionally we would get break-ins into the clinic, and they were always after a couple of things. One is the narcotics that we would give animals for things like pain control. But another thing they would steal was horse anabolic steroids and sell them on the black market. So just a warning about the abuse of anabolic androgenic steroids. It can have things like cardiovascular disease and have numerous problems associated with heart function. And if you remember the trans fats and saturated fats and how they can change the ratio of LDL and HDL. So recall LDL, low density lipoprotein. So it contains a steroid group, and so low density lipo, and it contains a protein group or a polypeptide group. LDL is the bad cholesterol, and you want to have a ratio of high LDL to low LDL. And unfortunately, the abuse of anabolic steroids causes the ratio of LDL to increase compared to HDL. Other problems are psychological in nature, so things like aggression, they call it roid rage, and psychotic episodes, anxiety, hair loss. And that hair loss is not only irreversible, but it can also happen in females when you have the male pattern baldness. The male pattern baldness is based on the levels of testosterone, and as testosterone increases in women who abuse anabolic steroids, the male pattern baldness will often arise, and again, completely irreversible. And another thing with the abuse of anabolic steroids in females is the virilization, where the women will have a lowered voice. And of course, acne is another side effect. But there's more. In males, it can result in infertility, and gynecomastia is male breasts, which are, again, are irreversible. When I saw this photo, when I was talking in class, I was like, why are there raisins on here? Because I had put it in the semester before and I had forgotten about it. And it's a representation of one of the side effects of males, which is testicular atrophy, which means that it causes the testicles to basically shrink into little raisins. Last semester, it was so funny. In the chat, somebody put, so if you eat too many raisins, will you get testicular atrophy? Which I thought was really funny. In women who abuse steroids, they can have the reverse. Instead of gynecomastia, they can have breast reduction or breast atrophy, and all sorts of disorders associated with menstruation, and the permanence, as I mentioned, of a lowered voice. As a boy, when he goes into puberty, will lower their voice, have a lower voice, and it will also result in facial hair in females that abuse anabolic steroids, primarily testosterone. I watched an interview one time with a woman who this was back when there was East and West Germany, and she was competing in the Olympics for East Germany. And she and teammates were encouraged strongly to take anabolic steroids to make them better performers. And when she was talking, you could hear that lowered voice. And again, it's permanent. Don't abuse steroids. So phospholipids are often represented simply as having a hydrophilic head group and two hydrophobic tails. It's easy to forget, what is this hydrophilic head group and what are these little squiggles? The hydrophilic head group is actually a phosphate group that has other molecules attached to it, and the two squiggles are not flagella. They are each fatty acid tails. And the fatty acid tails, there's two of them, and the phosphate group with its attachment are all held together by a 3-carbon backbone, which is called glycerol. And think to yourself, what other molecule have we talked about that has a glycerol backbone with fatty acids attached? So waxes are the last type of lipid. recall that we've done other types of lipids. We already did the fats and the steroids and the phospholipids and finally the waxes. They have something that you've seen before, a long hydrocarbon chain that had a carboxyl group associated with it. So this is a fatty acid chain, which this carboxyl group was a reactive group that served as an attachment point for a long chain hydrocarbon with a hydroxyl group. Okay. So this is a long chain because it has a hydroxyl group. They called it a long chain alcohol. And this is highly hydrophobic, a carbon-hydrogen skeleton. Here you have this that has a double bond. And waxes, as you know, it can be structural. So beeswax and candles and plants will make waxes. And this is showing that the waxes will keep the moisture from being lost from apples, and it will also can protect from insects. So lots of functions of waxes, and not to mention the waterproofing that occurs in nature for the plants, and things like birds have waxes on their feathers. Here's a little video from my travels to the Galapagos. These are frigate birds, and they hunt in the ocean, and they get a lot of salt water. And they will wash their feathers in this freshwater lake. And so they're not catching fish here, they're actually using the fresh water to remove the salt. The problem with these poor things is that they lack lipids on their feathers, and so they can only skim the surface of the water and have to fly off. And they have to be careful because if they get too deep, they will get stuck. Luckily, this bird got away. And it's not lipids if we don't talk about the misery of lipids, which are pimples. And as mammals, we have sebaceous glands, and the lipids that the sebaceous glands will secrete is called sebum. And sebum has a bunch of different lipids that it is composed of. Here's a hair follicle, and there is bacteria that live on the surface of our skin naturally. One of them being Propionibacterium acnes. And the bacteria is fine until it gets, the hair follicle gets plugged with things like the sebum and the cells that make up the hair follicle, and the bacteria will get trapped inside of this plugged region of the hair follicle, and it will attract the white blood cells that are attracted to the bacterium that is in this plugged follicle, and the white blood cells will then cause the formation of this pimple. So, sorry about that. And that is it for the lipids.